Hello, this is Dr. David Brady, and welcome to this short course on the gut autoimmunity axis, insights into microbial influence. My disclosures include the fact that I am the Chief Medical Officer and one of the founding partners for Diagnostic Solutions Laboratory, or DSL, in the laboratory space, and also the Chief Medical Officer for Designs for Health, Inc. in the nutraceutical and nutritional supplement space. I've been practicing functional and integrative and nutritional medicine for over 30 years and spent 25 years in medical academia at the University of Bridgeport. There's simply no doubt that no matter how you look at it, there's an incredible rise in the incidence of immune regulatory disorders or autoimmunity. It spans across many different subtypes of autoimmune disorders affecting many different tissues. And if you look at these epidemiological incidence curves from 1950 forward, you'll see a meteoric rise. While autoimmune disease certainly involves genetic propensity, certainly the genes do not change this quickly. There is obviously an environmental factor interacting with the genetics of a person who may be afflicted by autoimmunity. When epidemiologists see curves rising this quickly, one of the first things that usually comes to their mind is an infectious epidemic. And we will actually look at autoimmunity in this short course as an infectious disease, looking at various pathogens and opportunistic organisms with an eye toward the GI microbial composition as potential factors and perpetuators of an autoimmune process. In order to properly look at this and frame it, though, we have to look at illness in a different way, a more holistic, a more networked way, a systems biology way, if you will. And that is what functional medicine strives to do. And autoimmune disease is certainly no exception. So let's look at autoimmune disease from a bit of a different perspective and see if that may yield us any potential to intervene therapeutically in ways that would move the dial on patients. If you look at this diagram, it's pretty much a map of how medical students, and probably all of us, learned immunology today. Very granular, looking at all of the different white blood cells or leukocytes and their various subsets, and the various metabolites and compounds and signaling molecules that they may manufacture and excrete or degranulate and exposed to the system. These include things like cytokines, interleukins, and so forth. And while it's very beneficial to understand these different types of very detailed issues of immunology, and it certainly informs mechanistic research and even research looking for therapeutic agents, it really doesn't help us that much with a patient in our office the next morning who has an autoimmune disorder and wants us to do something about it. Sometimes you have to simplify it and climb upstream and look at foundational reasons why someone may be susceptible to getting ill in the first place, and basically lose all of the granular details temporarily while you focus on sort of foundational big ticket items that are actionable that the patient can do that may actually change their outcome. It's certainly no coincidence why we would look to the gut when we're trying to figure out what may be stimulating an autoimmune response or a dysregulation in the systemic immune system. The dominating part of the immune defense is localized in the gut, and it's estimated that no less than 75% of the immune cells of the body are suggested to be found in the GI tract. Mucosal immunity is of prime importance, and it may very well be where the dominoes start to fall in the majority, maybe not the entirety, but the majority of autoimmune illness. This is a modern thought process or application of philosophy and ideas that were brought forth a century or more ago by people like E. Mechnikov, the famous Russian pathologist and zoologist, the 6th ever Nobel laureate in 1908, and the father of the orthobiosis theory and the father of modern probiotics. And while Mechnikov did not have the benefit of all of the modern understandings that we have, and certainly was not even able to really understand or ascertain what type of organisms were actually present in the human GI microbiota, today we have the benefit of the learnings from the Human Microbiome Project, and we are starting to better understand the interface between human health and or disease and the microbiome in general, and specifically with what we're talking about today, the subset of the microbiome, which is the GI microbiota composition. The associations, the connections, and even causal mechanisms have been discussed very thoroughly in the medical literature, and there's more coming out every day. We've probably just scratched the surface in understanding these connections. But one person who's done tremendous work, of course, is Yehuda Schoenfeld in Israel and his group, in looking at the microbiota at the crossroads of autoimmunity. And leaving all of the intricate details aside for a moment, this just makes intuitive sense. We have this hollow tube running through the middle of our body that is technically outside of our body. It's a tube that's filled with all kinds of things that can be very deleterious to us if they were allowed to interface with us systemically, so into our actual bloodstream or lymphatic system. And that includes a large number of various microbes of different types, including bacteria, fungi, protozoa, helminths, viruses, phages, and others. Many of these would not be healthy and can actually be lethal very quickly if they were exposed to systemic circulation. We also have this tube running through the middle of us, being constantly filled with food that we eat that is full of proteins and then broken down into various peptides that would be considered foreign to us by the immune response and can be highly antigenic. There's also a tremendous number of toxins that's produced as we digest and metabolize our food. And the only thing separating us from all of that nastiness is a one cell lining thick mucosal lining or barrier. So we will talk a lot today about the composition of these microbes and some of their metabolites and how they can interact with our immune system, both locally in the gut with mucosal immunity and also systemically. Particularly systemically if there is barrier integrity issues or so-called intestinal permeability and or leaky gut. An example of this that was published in Nature and then ended up in a piece in The Scientist is an interesting study where they took wild-type mouse and they introduced one bacterium into the mouse's GI microbiota, and that was H. hepaticus, or Helicobacter hepaticus, same genus, different species than the well-known potential ulcer bug, Helicobacter pylori. And they basically induced or created an experimental mouse version of the autoimmune disorder inflammatory bowel disease. By introducing one bacterium in H. hepaticus, they highly activated Th17 cells, which release cytokines associated with inflammation, such as IL-17, causing the symptoms of inflammatory bowel disease. Then they introduced one more bacterium, B. fragilis, which expresses a polysaccharide A or PSA to the dendritic cells, which take it up and present this PSA molecule on their surface, which in turn activates CD4 or Treg cells. And this causes a release of the anti-inflammatory interleukin 10, which suppresses the inflammatory action of the IL-17 first induced by H. hepaticus, alleviating the IBD in mice. Now, why is this important? Because they took a healthy animal population, they changed one organism in the GI microbiota, they created inflammatory bowel disease, an autoimmune disorder, and then they essentially resolved that disorder by introducing one more bacteria, which influenced mucosal immunology. So this is really profound. And are there lessons here for the human experience? Yes, there are. This was a perfect experiment to show how the composition of the GI microbiota can alter the balance between anti-inflammatory and pro-inflammatory states in the gut, and the balance between immune tolerance and immune aggressiveness, which can ultimately take the form of autoimmunity. We've often learned about Th1 dominant versus Th2 dominant autoimmune disorders, and that usually there's an imbalance in the Th1, Th2 activity of the immune system systemically in an immune dysregulatory disorder. But when we come down to the gut environment and mucosal immunology, the analogy is an imbalance between the Th17 aggressive cells and the T regulatory cells, which calm down things and create immune tolerance and move us away from an inflammatory posture. Ways in which we can use lifestyle medicine, whether it's dietary, whether it's nutritional supplementation, whether it's botanicals, whether it's various other ways of altering the GI microbiota composition, we want to strive for this balance between Th17 and Treg cell activity. It's a yin and yang scenario here, and we want to find that balance to put the patient or subject in the best immune posture when it comes to intestinal immunology. As I mentioned previously, there's a lot of literature now looking at different autoimmune and systemic inflammatory disorders being associated with predictable alterations or imbalances in the GI microbiota composition or dysbiosis. And the additional wild card here is intestinal hyperpermeability. If someone has a dysbiotic state and an overgrowth of organisms which may not ultimately produce a favorable immune stance in the gut, if there is also a concomitant issue of intestinal hyperpermeability or problems in barrier integrity in the gut, it seems to really set the stage for autoimmunity and inflammatory systemic disorders, particularly in those that have predisposing genetics. An example of a very complex chronic disorder that is multifactorial and not classically autoimmune, but still a complex metabolic disorder, of course, is autism. And now with the availability of real-time quantitative PCR, we can really pick apart what the intestinal environment looks like, particularly the GI microbial composition in autistic subjects versus children who are not on the autistic spectrum. And there are interesting things that are found. Those on the autistic spectrum have a much higher representation of Clostridia in their guts. And there's various Clostridia clusters that have been elucidated in the literature. Now, what does this mean? Does it mean, aha, Clostridia is the cause of autism? No, autism is much more complicated than that. But can Clostridia overgrowth in the GI tract be one of the factors involved in creating a scenario that may ultimately express as autism? We don't fully know, and we don't even know if the Clostridia is a result of autism or if the Clostridia is contributing to autism or both. But it's interesting as we look further down this hole to find out exactly what are potential mechanistic understandings of why alterations in the GI microbiota may alter a subject toward a complex disorder such as autism. Studies have shown, and certainly experienced functional medicine clinicians know, that favorable changes in dietary intake that alter the microbiome in a positive direction, and things like conventional probiotics and prebiotics can certainly, at least functionally, help autistic spectrum disorder subjects to some degree. However, the most powerful therapy we have to date to alter the GI microbiota composition is fecal transplantation. And there are some studies, although limited, on FMT therapy in autism that have shown significant improvements with various functional assessments in autistic subjects. There may be no area of medical research that is hotter right now in producing more studies on an ongoing basis than looking at the GI microbiota and attachments to various diseases and disorders. The role of the gut microbiome in systemic inflammatory disease is a great paper to pull and read if you get the chance. This paper fully explores the different signatures, we call them, or states within the GI tract that may be novel or unique to subjects with specific medical conditions or diseases. And this does not only involve the alterations in the microbiotic composition. In other words, we can figure out which organisms may be overrepresented and underrepresented versus normal control subjects in a certain disease process, such as, let's say, rheumatoid arthritis. But the signature also includes the ramifications metabolically of that switch or of that alteration in composition, including how does it change short-chain fatty acid composition in the gut? How does it alter bile acid metabolites? What does it do to cytokine expression? What does it do to the balance of inflammatory versus anti-inflammatory cytokines and other inflammatory mediators? These are all things being hotly studied and understood and mapped, and this information is being used to try to develop therapeutic agents to treat these different medical conditions. An example of the development of therapeutic agents by the pharmaceutical industry to somehow affect the GI microbiota composition or the metabolic state within the gut can be found on this slide, including one of the items, symbiotics, which is actually a Bacteroides fragilis-derived polysaccharide A for the treatment of IBD and multiple sclerosis. And this is based on the study I showed you previously, where they introduced B. fragilis after inducing autoimmunity and inflammatory bowel disease in mice, and then were able to resolve it. This is a furtherance of this work, and this is in preclinical development as a therapeutic agent at this point. Over the last decade plus, we've had an evolution of stool analysis that's allowed us to be able to look very deeply into the GI environment, including very specifically the composition of the GI microbiota, and better understand what is actually there, what is not there in healthy conditions and in specific disorders. There's pretty much been an entire migration from conventional microbiology type of methods to molecular methods in stool analysis. But even within molecular methods where we're using the DNA or the genetics to determine what organisms are there or not, there are different types of molecular methods. The two major ones being quantitative PCR and sequencing. And we get some of the differences between those two and when each one may be more appropriate than the other in different situations. Let's explore some of the difference between quantitative PCR and sequencing molecular methods. Sequencing is not quantitative, it's relative. So it will give information about a large number of organisms that may be present, but only in relative populations to one another, not as absolutely quantitative amounts. It's also relatively or comparatively poorly qualitative. It's pretty much decent at getting down to the genus level, but not too great getting to the species level with any deal of fidelity. And going beyond the species level to strain level or toward looking at the genomics of metabolic characteristics, let's say, of different organisms within a sample is not really in the realm of sequencing. But where sequencing does shine is if you want sort of a 30,000 feet view of things. In other words, it's good when you're looking at large number of subjects and you're trying to determine exactly what may be there. So you're looking for everything that's there and the relative quantities of each, but not the absolute quantity of specific organisms. So it's relative versus quantitative amounts, and it's not medically diagnostic. For instance, it's not insurance reimbursable in a medical diagnostic setting for these reasons. It's really more used in research and then data crunching. So if you want to know the difference in the entire composition of the microbiota between people with rheumatoid arthritis and without rheumatoid arthritis, or people in Los Angeles versus people in Tokyo, or any other comparison you want to make, then doing some level of sequencing with the right number of reads and fidelity, which is usually done in academic research and rarely done in direct-to-consumer types of tests using sequencing, then sequencing and the data that's produced by it can be analyzed, and that would be the way to go. However, if you're a clinician trying to determine if there's an overgrowth of specific organisms, let's say pathogens or key opportunistic organisms that may be overgrowing, and if they're overgrowing, are they overgrowing to a point at which it triggers a medical intervention, then quantitative PCR is absolutely the way you want to go. This is just an example of a sequencing test where you can see, this is one page of a multi-page report, and many of you have seen these, and patients may have dropped them on your desk, and it's page after page after page of lots of organisms, many of which you've never even heard of, and you have no idea what their clinical significance is, because it's really not meant to be used in that type of environment in a patient-doctor interaction making a medical decision. These are things to be ascertained and crunched by computers and compared when looking at different types of study populations. So you can see here, I just put a little red circle around the ranges, and you see the ranges are not a quantitative amount, it's a relative percentage of the total DNA captured in the sample. So this is a relative abundance type of referendum, organism by organism, versus an actual quantitative amount. This is very different than looking at a report of a curated group of organisms that are determined quantitatively with qPCR, such as in GI map testing. On this one page here, you see pathogens reported, and they're reported at actual quantitative levels. And a reference range is provided to determine if those quantitative levels in the case of pathogens would be considered above the threshold where treatment would be suggested. You can see here two positive reports for Clostridia difficile or C. difficile, but not just that the organism is present, but that the organism actually contains the genetics to produce toxin A, and in this case toxin B, which are the common toxins to cause diarrhea, cramping, and pain, and the clinical symptoms associated with C. diff infections, often after antibiotic use. Also, on the GI map report, you will see conditional pathogens such as Helicobacter pylori. Not only is the organism's presence reported, and if so, quantitatively compared to a reference range, but also, again, you can dive deeper with quantitative PCR and determine if the genetics are there within the organism's population to produce the virulence factors that are what are responsible for erosion of the gastric lining, duodenal lining, and can ultimately produce ulcers and even higher incidence of gastric cancer. Also, looking further down in the report, you can get a referendum on commensal and keystone bacteria, the so-called good guys, the organisms that should be there and should do something for us as we provide them a home. So having a good commensal signature is very important, and it's associated with healthy states, where dysbiosis of commensal signatures is commonly found in diseased states. While quantitative PCR is used in conventional medical situations involving limited pathogens panels, the GI map test is actually using this type of methodology over a much larger curated group of organisms that would be of interest to the functional and integrative medicine practitioner. Not only overt pathogens, not only conditional pathogens, not only commensal or favorable organisms, but then everything else in between those two extremes, which is where most of the organisms really are. And these include opportunistic or overgrowth microbes that are usually the ones that overgrow when there's lack of abundance of the commensals. You will get a overgrowth of opportunistic organisms, and the overgrowth of these organisms are things that can contribute to more what are considered conventionally functional bowel disorders, such as irritable bowel syndrome. They can cause lots of symptoms like gas and bloating, distension, changes in bowel habits such as tendency toward constipation and or loose stools, or a variance between those two. And also there's curated groups of these organisms that are known to be inflammatory, that can stimulate autoimmune related activity. These would include inflammatory autoimmune related bacteria that would be considered just opportunistic, and even those that are commensal. Some of our commensals, when they overgrow, can be associated with higher incidences of autoimmunity. And then, of course, we can look at overgrowth of organisms which are known to produce a lot of gases and can be attributed with things such as small intestinal bacterial overgrowth, such as Methanobacteriaceae. And of course, quantitative PCR can be used to look at the remainder of parasites, including the smaller protozoal organisms and the larger helminth organisms. And then what is a real value added in a panel such as GI mapping versus sequencing, particularly direct-to-consumer sequencing, is you actually get diagnostic intestinal health markers or stool chemistries that will allow us to look at things like digestion, such as steatocrit and elastase for pancreatic exocrine output. Other GR markers, including is there blood, is there beta-glucuronidase that can deconjugate hormones, for instance, being eliminated through the bowel. Immune response markers such as total secretory IgA, anti-gliadin secretory IgA as a potential marker for non-celiac gluten sensitivity, markers of immune activation or white blood cell degranulation such as eosinophil activation protein, markers of mucosal inflammation such as calprotectin, and then markers for intestinal hyperpermeability or barrier dysfunction such as fecal zonulin. A final added benefit would be to be able to look across the GI microbiota population for antibiotic resistance genes. Therefore, if you're trying to intervene or need to intervene with antibiotic therapy, such as in the case of H. pylori infection, then you can pick antibiotics which would not induce further antibiotic resistance in that subject. With greater understandings of these associations between various microbes overgrowing and the likelihood or the presence of specific autoimmune disorders, it's very important to have a tool like GI Map to turn to actually make an individualized referendum on a patient's microbiota composition to determine if there needs to be action to improve their dysbiotic state and lower their inflammation, lower their immune agitation, and try to get them on a path to better GI health and better overall immune balance. And in doing so, we would really recommend that you particularly key in on these specific curated areas on the report, looking at microbes that have been associated with inflammation and autoimmunity specifically, such as organisms like Citrobacter, particularly Citrobacter frundi, Klebsiella, and particularly Klebsiella pneumoniae, Proteus, and particularly Proteus mirabilis, Prevotella, and others. This may trigger an intervention with natural GI antimicrobial options and volatile oils, which has been shown to favorably alter a dysbiotic state and actually be more functionally beneficial over the long term than more powerful immediate interventions like GI-specific antibiotics such as rifaximin and xifaxanide. Both can produce very good immediate symptomatic relief, but the functional approach with standardized botanicals and volatile oils tends to produce a longer therapeutic benefit and a stickiness of the treatment that you do not see when too many of the good guys are also killed with things like prescriptive antibiotic therapeutics. An example of one of these sample natural GI antimicrobials is shown here with a multitude of berberine-containing botanicals, citrus or grapefruit seed extract, caprylic acid, Juglans nigra, a very antimicrobial botanical, and even the botanical tribulus, which is a wonderful GI antimicrobial. The addition of a volatile oil such as oil of oregano can greatly expand the therapeutic effectiveness by covering a wider sensitivity spectrum of the type of organisms that we want to lower in the microbiota composition in dysbiotic states. Of course, looking at markers such as calprotectin and zonulin are very important to look for barrier integrity or hyperpermeability issues, which also would need to be addressed. And while overproduction of something like zonulin, which can relate to leaky gut, can be a genetic issue, making one more susceptible to autoimmune disease, the vast majority of instances, zonulin overproduction is a direct result of overgrowth of the wrong type of microbes, such as pathogens and opportunistic organisms that are related to dysbiotic states. Natural interventions to improve barrier integrity may include products which contain things like L-glutamine, which is the preferred fuel source for the enterocytes to keep them healthy, but also immune proliferation or immune activation blocking mucopolysaccharides such as N-acetylglucosamine, and also the use of classic mucilaginous botanicals that can do the same thing using glycomics in your favor to downregulate the immune response. These include herbs that sort of have a sticky, snotty sliminess to them, including aloe vera, slippery elm, marshmallow, chamomile, okra, cat's claw, all very, very therapeutic in this type of situation to improve barrier integrity and to lower immune amplification. And while there's many things that go into autoimmunity, not only the subject's genetic predispositions, but many different antigenic environmental agitators, if you will, including various dietary proteins, which we did not talk about in today's module, chemicals as well. But infections are definitely a big part of this. And it's not all GI microbiota. Certainly systemic viruses like Epstein-Barr virus, cytomegalovirus, HSV, and others are very in play in many autoimmune disorders. One source of microbial antigenic triggering of the immune system that can end up in an autoimmune state certainly is this massive reservoir of bacteria contained in the intestinal tract that, again, is a hollow tube running right through the middle of our body, but technically outside of our body. So I hope there's a greater appreciation for that after this module, and you can utilize very, very useful tools like the quantitative PCR-based stool analysis known as the GI map. More information and a deeper dive can be taken by reading some of my publications regarding this topic, including in the Open Journal of Rheumatology and Autoimmune Disease, and also some good ones in trade publications like the Townsend Letter. They're available on those publication sites, but you can also grab them in the articles tab on my website at drdavidbrady.com. I thank you for your time and your attention, and I hope it was worth your while. And please, if there's anything we can do in our clinical and technical and scientific support department at Diagnostic Solutions Lab to help you better understand these concepts, better understand how to use our testing, including the GI map, quantitative PCR analysis, and many others, please don't hesitate to contact us. Thank you very much.